All right. Hello, everyone. My name is Dr. Jeff Shu. I'm one of the assistant professors in cardiology. And I'd like to get started with today's seminar. And it is my great pleasure to welcome Dr. Tony Rosenzweig virtually to UCLA for our CTSI Distinguished Speakers Series. Dr. Rosenzweig is the Chief of Cardiology and co-director of the Corrigan Minehan Heart Center at the Massachusetts General Hospital, or MGH, as well as the Paul Dudley White Professor of Medicine at Harvard Medical School. A graduate of Harvard Med and the MGH Internal Medicine and Cardiology training programs, Dr. Rosenzweig has a longstanding interest in identifying new mechanisms and targets in heart failure, identifying novel pathways that are functionally important in the heart's response to both pathological and physiological stimuli. His lab has had a longstanding interest in understanding how exercise protects against cardiovascular disease. And these studies identified exercise as the only known physiological regulator of cardiomyogenesis in the adult heart, and went on to delineate some of the responsible pathways. He's co-authored close to 200 papers in leading journals such as Cell Metabolism, Nature Communications, Science Translational Medicine, and Circulation. His work is cited over 28,000 times with an H index almost as high as my credit score of 86. Dr. Rosenzweig has been particularly interested in the translational potential of these discoveries and is a scientific co-founder of a biotech startup focused on developing small molecule therapies for a genetic cause of sudden cardiac death, and has served as an advisor to pharmaceutical companies in the past. Importantly, Dr. Rosenzweig is also a committed mentor who has personally supervised over 60 fellows in his lab, the majority of whom have been physician scientists and some of whom I've had the privilege of working with over the years. Most who have left the laboratory have gone on to full-time academic faculty positions with substantial research commitments. And of these, 14 hold major leadership positions, including as center directors or department and division chairs at major academic medical centers and universities. In addition, Dr. Rosenzweig has previously served on the scientific board of the Sarnoff Foundation, an organization devoted to mentoring young investigators. And he's the program director of the MGH NIH T32 training grad, focused on training the next generation of cardiovascular investigators. Dr. Rosenzweig is a recipient of numerous awards, including the Roman W. DeSanctis Clinical Scholar Award, as well as an Outstanding Investigator Award from the National Heart, Lung, and Blood Institute. He's been elected into prestigious societies, including the American Society of Clinical Investigation, the American Association of Physicians, the Association of University Cardiologists, and the Association of Professors of Cardiology. So again, it is with great pleasure and excitement that I welcome Dr. Tony Rosenzweig to UCLA as our distinguished speaker for today's seminar, Linking Exercise to Cardiovascular Benefits. Take it away, Dr. Rosenzweig.
Great. Well, Jeff, thank you so much, first of all, for the kind invitation, and then for that overly generous introduction. I wish my kids were here and they would give you the counterpoint to everything you said, but really nice of you to say that. Now, Lucy, do you need to stop sharing so that I can share, or? Okay, great. So let me see if I can pull up my slides here. So Jeff, are you seeing that? Is that look good?
Yeah, looks
perfect.
Good.
Again, thanks again for the invitation. Really delighted to be able to visit with people. I had a wonderful morning and got to see some old friends as well as make some new friends, and really enjoyed hearing about the exciting things going on at UCLA. Again, sorry it wasn't in person. Hopefully there'll be another opportunity in the future. And I'm not just saying that because it was 34 degrees here this morning, but it would be great to see people in person and be able to connect. These are my disclosures. As Jeff mentioned, I am a co-founder of a company focused on developing small molecule therapies for long QT syndrome. Most of this is not relevant, although MGH, the last thing, has filed a patent on one of the topics I'm going to talk about today, this long non-coding RNA, and we're hoping that will move forward to translation eventually. So I know that this doesn't really require much introduction for this audience, but just briefly as a clinical context, we've obviously made enormous progress in the treatment of cardiovascular disease over the last 5 or 6 decades. This was from a review that Eugene Brownwald and Bessie Nabel wrote for the bicentennial of the New England Journal, just pointing out how the death rate has dropped so precipitously over this time period, and kind of linking that to some of the scientific innovation that has occurred over that time frame. But the other side of that coin is that despite this progress, the burden of cardiovascular disease continues to rise, as you all know, and this is just one metric, and probably not the most important metric. The cost to society in both direct and indirect dollars. This is from the American Heart Association, and I know you can't really see this fully, but projected out to 2030 is the last date there. The cost is estimated at $800 billion. So how do we understand this paradox that we've made progress, and yet the burden of cardiovascular disease only seems to increase? And I really think there are 3 principal drivers of that. The first is, in some sense, the burden of our success. We've gotten good at getting people through their heart attacks or treating their acute hypertensive emergencies, but then those people who survive, fortunately, go on and develop the late sequelae of cardiovascular disease, notably heart failure and arrhythmia. As you all know, we're in an epidemic of cardiometabolic disease, which we'll come back to a little bit, and that's a driver of all the metabolic component of that is a driver of all forms of cardiac and vascular disease. And then finally, our populations are aging both in the United States and around the world, and age is closely linked to cardiovascular disease. This is just showing the prevalence of cardiovascular disease as populations age in the United States for men and women. You can see that by the time people in their 80s, the large majority have some form of cardiovascular disease, although a lot of that is driven by hypertension and the revised definitions of hypertension. Still, when you look at hospitalization, for many years now, heart failure has been the leading cause of hospitalization in older adults. And when you look at heart failure, we like to say that about half the heart failure we see is heart failure with reduced ejection fraction, and about half is heart failure with preserved ejection fraction. But the rate of increase is greater for heart failure with preserved ejection fraction, HFpEF. And so the anticipation is that over time, this will become even greater. And as you all know, while we have a handful of different therapies that have been shown to be effective clinically for heart failure with reduced ejection fraction, there's really only one that has met its primary clinical endpoint for heart failure with preserved ejection fraction, SGLT2 inhibitors. That really just reduces hospitalization rather than, at least so far, being shown to improve survival or mortality. And even when you look at HFrEF, I guess I would argue that compared to our colleagues who are oncologists, where they have a real plethora of different therapeutic approaches that can be tailored to individuals based on the genotype of their cancers, we really have a relatively modest tool chest of things to deal with. And so there is a large unmet need, which is over time anticipated only to get larger for the reasons I outlined. So with that context, I think a number of years ago, we became interested in this idea that heart failure often develops after a period of abnormal growth called pathologic hypertrophy, shown here in this review by Joe Hill and Eric Olson, where the blue is meant to depict fibrosis, and you see thickening of the heart wall. And obviously, patients at that stage are at risk for heart failure with preserved ejection fraction. But some of those patients go on and dilate and develop heart failure with reduced ejection fraction, and both of those states are at risk for arrhythmic complications. But the heart also grows in response to other stimuli, more physiologic stimuli such as exercise, and in general doesn't lead to these adverse sequelae. So we became quite interested in what the difference was between these two growth states and why one would lead to these negative outcomes, while the other not only doesn't seem to lead to those negative outcomes, but actually appears to protect, at least in animal models and in some observational studies, and in rare randomized trials, against some of these cardiovascular outcomes. So I think when we started this, we really had sort of two fundamental questions about how these states of growth differed. And the first was, are they truly qualitatively different responses, or are they just quantitatively different? And so many people pointed out that, even if you're an elite athlete like Michael Phelps, you still probably exercise 4 or 6 hours a day. And then for the other 20 or 18 hours, you're resting. And maybe that period of recovery is essential in turning this stimulus, the physiologic stress of exercise, into a beneficial outcome as compared to people with aortic stenosis, where obviously it's present 24 hours a day, 7 days a week without any respite. So maybe these are just a question of degree. And the second is obviously exercise does a lot of things systemically. It changes your metabolism, it changes your systemic vasculature, it has profound effects on skeletal muscle depending on what kind of exercise you're doing. And that all contributes to its benefits. But are there also, we wondered, intrinsic cardiac pathways that are altered by exercise that are playing an important role in the benefits of exercise? And if there are, and if we could identify them, we wondered whether those pathways could be exploited, used in some way to either prevent or treat heart disease. So to summarize, really over a decade of work that people in the lab have contributed to, we set out trying to look at models of exercise in mice. And so we started with the swimming model, and to do this, you need a high-tech swimming pool, which was a bin from, I think, Target for about $4.95. You need to make sure the water is the right temperature. You need a good lifeguard, otherwise the swimming model turns into a drowning model, which is potentially interesting, but a separate question. And you can see that the mice are not necessarily natural swimmers, and some of them are either smarter or lazier than the others, and will kind of hang on to the side of the bin or the thermometer, and those are not so much the ones we're interested in. And this was a graded response, and so there's the lifeguard rescuing one that was struggling a little bit to avoid that drowning model. And so we started with 10 minutes twice a day, and then ramped up by 10 minutes for each session, so that by the end of 2 weeks they were swimming about 90 minutes twice a day, or 3 hours of swimming a day. So this is pretty intense. But I've got to say, over time, we've found that almost all the things we find in that model, although there are the differences in the molecules mediating them, we can recapitulate with a much less stressful voluntary wheel running model shown here. If you give mice a wheel, they will run typically 5 to 7 kilometers a night. And we also prefer this model because it doesn't disrupt their circadian rhythms, since mice are nocturnal. So to summarize sort of 3 key findings over the past decade or more of work that were at least surprising to me. The first is, when we look at the pathways, whether that's transcriptional profiling for messenger RNA or non-coding RNA or proteomics or post-translational modifications of proteins, when we look, there's really remarkably little in common between the exercise-induced cardiac growth and the pathologic, in this case, aortic constriction-induced growth, even when we looked really early, like 2 weeks in, when the hearts look identical. So the pathologic model hasn't led to fibrosis or apoptosis or dysfunction at this point. And so the hearts look identical, they've grown a similar amount, but the pathways are almost completely different. And when we find, not absolutely, but to the large majority, they're different. And when we find things that are in common, most commonly, if they're changed significantly in both models, they change in opposite directions. So while this can't really answer the philosophical question that I posed first, it does, in my mind, reinforce the idea that these really are distinct processes from an early stage, even when superficially the hearts look similar functionally and anatomically and pathologically, but there's something on the molecular level that has already set them down on 2 different pathways. The second thing that was interesting to me, and I think was one of our hopes and hypotheses, but really was not based on a lot when we started, was that in virtually every case, the pathways that we identify or others identify that are functionally important in the heart's response to exercise also protect the heart against pathologic stress, meaning that if we identify something, whether it's AKT activation or PI3 kinase activation or some of the non-coding RNA pathways, almost always when we mimic the change that you see in exercise in those pathways, you also protect against heart failure, ischemic injury, other pathologic models. So these 2 themes, in some ways, encouraged us to study exercise more, because they suggest that when you study exercise models, you find different candidates than the candidates you would find if you just studied the disease models. And secondly, the pathways and candidates that you find are enriched for things that are also protective against disease. So meaning you identify a set of things that are enriched for therapeutic targets. And there's also an advantage here that I just want to stress that again, you almost always mediate the benefit by mimicking the effect you see in exercise, whereas in the disease models, we find it more evenly split. About 50% of the time, the change you see is something that's compensatory and beneficial, and about 50% of the time, it's actually maladaptive. So you have less sense of what the directionality would be. And then the third thing that has emerged as a theme from these studies, which again was not one of our a priori hypotheses or something that I was imagining, but has turned out to be quite interesting, is the observation that very consistently when we did these kinds of genomic profiling experiments, exercise activated pathways in the heart that were linked to cell cycle progression, cell proliferation, or regeneration. Not so much in the heart, because the heart, as you know, has a very limited capacity for that, but in other systems. And it made us wonder whether, in fact, exercise might be doing something like that in the heart, and we'll come back to that. As you all know, there's been a lot of controversy over cardiomyogenesis in the adult heart, and whether there are resident stem cells, and whether the new cardiomyocytes are born at all, and to what extent. And I think my own take of the best evidence, both in animal models and in people, is there is some capacity to grow new cardiomyocytes. It's just very limited. And so the best data in humans comes from this science paper from Jonas Friesen and colleagues at the Karolinska over a decade ago, where they used the fact that people who were alive during nuclear testing were exposed to very low levels of radioactive carbon, labeling that they breathed in as carbon dioxide. If they were synthesizing DNA or cells were undergoing mitosis, they incorporated that into the DNA. And then in an autopsy series of people who died of non-cardiac causes, they were able to date the birth of the cardiomyocytes, knowing when those people were born, and show that there was an ongoing birth of cardiomyocytes in those people. In a healthy 20-year-old, it was about 1% a year, and then it becomes lower with age. Remarkably, that 1% is very similar to the rate that has been measured by Rich Lee and other people in mice, and much more conservative than some of the earlier estimates from other groups that have really largely been discredited. But I would keep that number in mind, that there's about a 1% birth of cardiac myocytes. And of course, from a clinical and scientific perspective, a really interesting question is, what regulates that? And is there some way to understand what controls it and potentially use that low level and turn it into something that might be more clinically meaningful? So at least as of the time that we started doing these studies, it really was completely unknown what the physiologic regulators. And so as Jeff mentioned briefly, we collaborated with Rich Lee, who had established a very elegant mass spec based system for really determining whether cells had undergone a mitotic event. And basically, this is using a quantitative mass spec system that uses stable isotopes. So giving mice, either through infusion or ingestion, N15, so the stable isotope N15 thymidine, as opposed to the more common N14 thymidine, and then being able to measure incorporation of that N15 thymidine. And the real advantage is that, first of all, you get this remarkable resolution shown here, so you can identify cardiomyocytes unambiguously, but then in addition, you can make quantitative measures. And so Rich was able to show in a Nature paper some years ago that he was able to distinguish, for example, DNA repair or even subsequent cell divisions of those cells, because they would dilute the amount of stable isotope labeled DNA. And so here you can see that you can quantitate and identify cells. So this cardiomyocyte in the upper right panel, you can see the colorful cardiomyocyte nucleus that has incorporated a quantitative amount of stable N15 thymidine, showing it's gone through a complete mitotic event, not DNA repair, but the nucleus on the right doesn't light up because it didn't go through this. And so we meticulously examined this in mice that were allowed to run on wheels for 8 weeks. And this is work that Carolyn Lurkenmuller in my lab did with Anna Vujic in Rich's lab in a collaboration published a few years ago. And interestingly and remarkably from my perspective, allowing mice to run, adult mice to run, these were young adult mice, increases the rate of cardiomyogenesis about 5-fold, so 4.6-fold over 8 weeks, shown here. So it takes that basal rate of about 1% and brings it up more to sort of a 5% annualized rate, which I thought was pretty interesting and remarkable. Now, what we don't know is, if you are a habitual runner, if you're a lifelong runner, is this cumulative? Are there adaptive mechanisms that come into play that maybe diminish the effect over time? But if this is cumulative, it would suggest that this could have a material effect in that balance between the homeostasis in the adult heart. More recently, we've actually looked in aged mice, so mice on the order of 20 months old, and shown that there's a similar phenomenon that is still possible even in very old mice. And actually, you can see here that the fold increase, if anything, is perhaps even larger because there are so few cardiomyocytes that undergo mitosis in sedentary old mice, although the final result is that the mice that are, first of all, I would say the aged mice run less than their younger counterparts. And then secondly, with running less, they also have, the exercise gets them back to what is comparable to the baseline of sedentary young mice. So I guess good news, bad news for those of us who are closer to old mice. It doesn't get us off the hook. We have to still exercise because there is some benefit, but you're sort of running fast just to stay in the same place that you might have been at age 20 or so. And of course, so we're quite interested in understanding what the mechanisms are and learning whether we could use those mechanisms in a clinically relevant context. And in that arena, we've been quite interested in the role of non-coding RNAs, and I know we have some real experts here, so this is certainly a bit of bringing coals to Newcastle. But as everyone here knows, much of the DNA is transcribed, but only a small amount of that, shown here 1.5%-ish, it makes protein. A lot of the other RNA that's made, has really been kind of more interesting and mysterious in some ways. And broadly, we think of this as small RNAs like microRNAs or small interfering RNAs, for which the Nobel Prize was awarded in 2006, and long non-coding RNAs, more than 200 nucleotides, which are, I think more challenging in some ways because they have they're less conserved than microRNAs. They can work through a number of different kinds of mechanisms, binding proteins that are transcription factors, binding other nucleic acids, including microRNAs, and acting as sponges. And so quite interesting, but both of these potentially control whole programs of gene expression, and so are quite interesting in the context of thinking about the response to physiologic stress. The other thing that's interesting is, of course, we've discovered, not we, but others have discovered that the field has discovered that they circulate, and some of them are in vesicles or microvesicles or exosomes. Some of them are bound to proteins. And I think we're still understanding what those mean. There's some evidence that at least in some cases, these can be taken up by distant tissues or cells, and that can be a form of communication that actually has some biological impact. But at the very least, they may function as biomarkers that could give clinically useful information. So in that context, we had looked a number of years ago at the microRNAs that change. And here we looked at the swimming model and the running model, and looked and focused in on the microRNAs that were in common between those. And one of the ones that was particularly interesting to us was this microRNA miR-222. Partly it was increased in exercised hearts, and when we mimicked it or overexpressed it in cardiomyocytes, there was sort of a dramatic downregulation of pathologic markers like AMP and BMP, and upregulation of physiologic markers like myosin heavy chain. And then interestingly, our colleague independently had shown some years ago that in young healthy athletes, miR-222 goes up in the plasma with or serum, I think it was, with exercise, both acutely and with chronic training. And then we showed that in heart failure patients, it also goes up. Not, of course, proving that it comes from the heart where it goes up in mice, but at least suggesting there's some conservation and resonance of the fundamental biology. So we were interested in understanding what miR-222 was doing, and this is published, so I won't dwell on it, but it works through a number of direct targets, some novel kinases, as well as some transcription regulators and the cell cycle regulator P27. And we were able to show that when you inhibit miR-222, there's really you lose the physiologic growth of the heart in response to exercise, shown as heart weight relative to body weight or tibial length on the right hand side. And so when we looked at miR-222 in the context of exercise-induced cardiomyogenesis, we were also able to show that if we inhibit it now with a locked nucleic acid modified oligonucleotide that brings down miR-222 in the heart about 95%, we completely block the induction of cardiomyogenesis as well. So we were quite excited and interested in this. And if you overexpress miR-222, you actually protect against adverse remodeling after ischemic injury, shown here with less dilatation and less dysfunction and a dramatic decrease in fibrosis, but not shown here when we overexpress it in baseline hearts without a stress or perturbation. There's no growth in the hearts. There's no markers or signs of cardiomyocyte proliferation. And so miR-222 is necessary for physiologic growth and exercise-induced cardiomyogenesis, but it's not sufficient. And so this launched us into trying to understand what might be sufficient, what are the pathways that really might be able to regulate this or recapitulate those benefits of exercise, which I'm going to spend most of the time today talking about. And this is work that really has been led by Habo Lee, who started as a postdoc in the lab and now has become an instructor and really has done a phenomenal job leading this project. So he started out by looking at mice that had been either exercised compared to sedentary, and then compared those to aortic constriction. These mice were run for 8 weeks, partly because we knew that 8 weeks was a time point where we had robust induction of cardiomyogenesis. And he identified 25 different long non-coding RNAs that were differentially regulated by running for 8 weeks. Again, these are young adult mice. And he termed these link exacts for long non-coding exercise-associated cardiac transcripts. And you can see here the kind of the Venn diagram. Once again, consistent with our overall theme, the things that we see in pathology, either TAC that short-term induces left ventricular hypertrophy or TAC that long-term induces heart failure compared to exercise, are largely distinct, so different. There were 6 that were statistically significantly changed in both the physiologic and the pathologic models. And interestingly, in every one of those cases, they change in opposite directions. Again, reinforcing this idea that these are really different. And so even when you find common pathways, they often are doing different things or changing in different ways in those 2 different models. And this is just a qPCR validation of kind of our top 3 candidates. And of these, the one that we thought was most interesting to explore in more detail is this one that we called Link Exact 1. And the reason we chose that is because there are several reasons, but one of the things that intrigued us was, as you can see here, Link Exact 1 decreases in exercised hearts and then increases in the pathologic models, both in left ventricular hypertrophy, pathologic left ventricular hypertrophy, and in heart failure. And so, again, our experience had taught us that, in general, mimicking the change in exercise is how you get the beneficial effects. And so this was uniquely the only one that went down in exercise of these ones that were in common, changed in both. And so we thought, if the hypothesis is correct and this is a therapeutic target, there might in fact be a practical way to inhibit this, because, as you all know, the field has had some success with a variety of inhibitory approaches to inhibiting RNAs. This is shown a New England Journal paper for amyloidosis, transthyretin amyloidosis, but a number of different targets have been successfully targeted this way. And obviously, this is one of the approaches we're using with these modified oligonucleotides to inhibit in these animal models. So the thought was at the outset that perhaps if this really did have a functionally important role, it might be a tractable target. Of course, one of the challenges, as I mentioned, about long non-coding RNAs is that they're often not highly conserved. And another thing that was appealing about Link Exact 1 is that when we cloned the full-length human clone, it's about 70% conserved at the sequence level, which is pretty good for these. And then in collaboration with Mike Bristow at the University of Colorado and Soumya Das at MGH, we've looked in both human cardiac tissue and human plasma. And interestingly, in both cases, as in the animals where it goes up in the hearts of mice with pathologic hypertrophy or heart failure, it increases in the heart tissue from patients with idiopathic cardiomyopathy who went to transplant. And in these human samples, these were plasma from patients with both HFpEF and HFrEF, and it significantly increased in both of those. So it increases in both those populations and doesn't seem related to ejection fraction, but I think of it more as a marker like BNP, which might be increased in both of those populations. Okay, so functionally what's it doing? And as always, we start with in vitro studies, but in the interest of time, I'm going to kind of hone in on the in vivo studies. So the first thing we did was we made an AAV9 vector with a cardiac, has some cardiac tropism, and then also engineered a troponin cardiac specific promoter and overexpressed link exact 1 in the hearts of normal wild type mice. And the level of expression we're getting is roughly comparable to what we see in some of the heart failure and pathologic hypertrophy models. And so just overexpressing in normal unperturbed mice, link exact 1 leads to an increase in heart weight and relative wall thickness, and you can see on the bottom in the middle there, pretty dramatic increase in cardiomyocyte size, no increase in markers of proliferation, and a modest decrease in fractional shortening, a modest increase in lung weight, suggesting there's some congestion, but I would emphasize those are modest compared to the degree of cardiomyocyte growth. And a pattern that you see here with increases in AMP and BNP and beta to alpha mice and heavy chain, and a decrease in PGC1, that all consistent with a pathologic model. So simply overexpressing link exact 1, which increases in human and experimental heart failure and hypertrophy, is sufficient to induce what I would say is a pathologic hypertrophy model in mice with some modest degree of dysfunction and congestion, suggesting to me anyway that probably the pathologic hypertrophy is the primary and dominant effect. Well, what about when we inhibit it? And so in these studies, we then used a modified oligonucleotide sequence, a specific locked nucleic acid that reduces link exact 1 in the heart by about 2-fold, which is very similar to what we see in exercised hearts or hearts from mice that had exercised. And the surprise was that while overexpressing increased the size of the heart, so did inhibiting it. So if we inhibit link exact 1, we also get an increase in heart size shown here and relative wall thickness shown there and cardiomyocyte size. But the difference is we don't see a pathologic pattern. So ANP and BNP and beta to alpha mice and heavy chain ratios don't change. And in fact, we see physiologic markers like PGC1 that goes up, and I'm just showing a few of the sentinel things here. Fractional shortening marginally, but actually significantly increases. There's no increase in lung weight, so there's no congestion. And in contrast to what I showed you with overexpression, we actually see an increase in markers of proliferation like EDU incorporation, Ki67, or phosphohistone H3, suggesting that in many ways, inhibiting link exact 1 recapitulates a lot of the phenotypes of an athlete's heart or an exercised heart. What about protection? So if we tack those animals with inhibition of link exact 1, you can see that they have less of an increase in heart weight. They have better preservation of fractional shortening. They have a significant 2-fold decrease in fibrosis and less increase in cardiomyocyte size. Again, emphasizing this idea that physiologic and pathologic growth of the cardiomyocyte may be in some dynamic tension, and they have some increase in these markers of proliferation shown here. Well, recognizing that aortic constriction, I think, is a useful model, but it doesn't really recapitulate anything we see clinically. Patients don't suddenly develop aortic stenosis. We looked at a number of other models, and shown here is the mice in heavy chain F764L mice that the Sibin lab had developed. This models a genetic cardiomyopathy that people get. These mice were over a year old when we looked at them. And so, I'm not shown here, but link exact 1 does increase in the hearts of these mice. When we inhibit it, we actually see, even in these relatively old mice, over a year old, an improvement in fractional shortening. So this is now delivered in a clinically relevant time frame. And in an ischemic injury model, if we deliver it after reperfusion, again, a clinically relevant time frame, not surprisingly, there's no effect on the initial dysfunction or infarct size, but then they start to separate over time. And the mice in which we've inhibited LINK-exact1 have a recovery of function associated with, again, about a twofold reduction in fibrosis at this late time point, a reduction in apoptosis of cardiomyocytes, and an increase in, again, markers of proliferation shown here. Well, how is LINK-exact1 acting? Well, I mentioned earlier that long non-coding RNAs can act through a number of different mechanisms, one of the things that they are known to do is to bind microRNAs and act as kind of a sponge, inhibiting the effects of those microRNAs in some instances. And so we looked in bioinformatic analyses at what was predicted to bind to LINK-exact1, and we're interested and encouraged to see that one of the predicted binders was our old friend miR-222. And so when we looked experimentally, miR-222 was actually the only one of the predicted ones that specifically bound to the sense strand of LINK-exact1. And when we mutated the putative binding site, we lost the activity. And not shown here, we did do some complementation studies in vitro showing that miR-222 does seem to have some effects downstream of LINK-exact1, mediating some of its biological effects. And so the model would be that LINK-exact1 increases and it binds miR-222, and so you lose the beneficial effects of miR-222. But we already know that miR-222 isn't sufficient to really recapitulate an athlete's heart. When we overexpress miR-222, which we've done in multiple inducible transgenic mouse lines, we don't increase heart size, we don't increase cardiomyocyte size, we don't increase markers of cardiomyocyte proliferation. So there must be other important targets. So one of the other things that long non-coding RNAs do is work through regulating transcription of other genes, sometimes genes that are nearby them. And so we looked, and the closest gene in both humans and mice to LINK-exact1 is something called DGSOM, which had not previously been annotated in the heart to have a functional role. And so when we looked in both human samples and in the mouse samples, there's actually a very good correlation. So settings where LINK-exact1 goes up, like heart failure, these are those human samples again, you see that DGSOM goes up. Similarly in the mouse, DGSOM goes up in heart failure, it goes down in exercise, it goes up when we overexpress LINK-exact1 and down when we inhibit LINK-exact1. So at least creating a circumstantial case that it's possible the two correlate, and when we manipulate LINK-exact1, we get effects in DGSOM that LINK-exact1 could in fact regulate DGSOM transcriptional levels. And in fact, when we then did promoter analyses, that is in fact what we see here. So you can see that when you have between 1,000 base pairs and 1,500 base pairs of the promoter of DGSOM, LINK-exact1 overexpression actually increases it, and knockdown modestly decreases it because the baseline is pretty level, and we can show that it directly binds to those promoter sequences. So LINK-exact1 does appear to directly regulate transcription of DGSOM. And it appears that regulation is important to its effects on cardiomyocyte proliferation. So these are now neonatal cardiomyocytes. So, obviously a simplistic model where it's easy to get them to proliferate. But you can see that if we manipulate the two together, it appears that knocking down DGSOM is an important mediator of the effects. And similarly down here, you can see that the induced expression by LINK-exact overexpression of AMP, BMP, beta-myosin heavy chain, the reduction in PGC1, and the increase in CBP beta, all markers of pathologic hypertrophy are mitigated by simultaneously knocking down DCHS2, suggesting that's an important part of this. Well, in Drosophila, DCHS2 has been reported to regulate hippo-YAP signaling, obviously one of the best documented regulators of cardiomyocyte proliferation. So we wondered if there was a hippo-YAP connection to exercise. And so when we looked at exercised mice, you can see that the cytoplasmic phosphorylated YAP decreases with exercise, and the total YAP in the nucleus here actually increases, suggesting there would be more transcriptional activity of, this is YAP1 actually. And similarly, when we inhibit a link exact one with a gap mirror modified oligonucleotide, you can see that we increase the nuclear total YAP. Conversely, in the heart failure models, when we subject animals to aortic constriction, there's more phosphorylated YAP in the cytoplasm, less in the nucleus. And similarly, if we overexpress link exact one, again, mimicking the change that you see in heart failure, we recapitulate that. We increase DCHS2, not shown here, but we increase the cytoplasmic YAP and decrease the nuclear total YAP. And then in cardiomyocytes, shown here, a lot of the downstream targets are changed again in the expected way. You know, we've done both the gain and loss of function complementation with DCHS2, which makes sense, suggesting that link exact one likely works through DCHS2 to regulate hippo and YAP, and that may well be an important part of the proliferation effects that we see. Well, does DCHS2 do anything in vivo in the heart? And so for this, we collaborated with Juan Ma Gonzalez Rosa, who's a faculty member at MGH, and Sam To, who's a technician in his laboratory. And they kindly generated cardiomyocyte-specific overexpression models of DCHS2, just shown here in green. And so the first thing is that overexpressing in the zebrafish model is sufficient to induce cardiomyocyte hypertrophy. Shown here again, is cardiomyocyte size increases and markers like ANP and BNP increase. And then if you subject these zebrafish to an injury model, and obviously as people know, one of the remarkable things about the zebrafish is its ability to recover from cardiac injury, whether that's cutting part of the apex, as Mark Keating and Ken Poss showed many years ago, or as Juan Ma has pioneered using a cryo-injury model, the hearts actually recover without scarring. But if you overexpress DCHS2, there's significantly less cardiomyocyte proliferation shown here, and the result is a much larger fibrotic area. And again, this correlates with changes in total nuclear YAP and cytoplasmic phospho-YAP. What about in mice? So zebrafish have this remarkable ability that if we overexpress DCHS2, we block. In mice, they don't have that ability. What if we inhibit or knock down DCHS2? So we used a Cas9 mouse to rapidly develop with viral delivery of guide RNAs, a knockdown model. And so with two different guide RNAs, we can get about a 50% reduction in DCHS2, which results in some hypertrophy, shown here an increase in cardiomyocyte size, again with a physiologic pattern of expression and modest improvement in function, and not shown here actually, but this also protects in more recent data against disease models like aortic constriction. And then just to show that this correlates with these markers of cardiomyocyte proliferation, again, in the mouse model. So we can recapitulate this with either inhibiting link exact one or inhibiting knocking down with these Cas9 mice, DCHS2. We started out talking a little bit about the importance of aging in heart failure and heart disease more generally. And just as a promissory note, I would say we've started to look at link exact one, and it does increase in hearts as mice age progressively, as does DCHS2. And when they exercise, it goes down. And so we're just trying to understand what the full functional importance of that may be. So to summarize, I guess I would say what we think is interesting about this is that link exact one and DCHS2 seem to be sort of at this pivot point between pathologic hypertrophy and heart failure on one side, and physiologic growth of the heart and protection on the other side. And so when link-exact1 and DCHS2 go up, as occurs in pathologic states, shown in the cartoon at the bottom, we think a couple of things happen. One is that there is more binding of miR222, and you lose the beneficial effects of miR222, but then there's also more transcription of DCHS2, which results in phosphorylation of YAP1 and sequestration in the cytoplasm, so less of a drive for repair and proliferation. Conversely, when link-exact1 and DCHS2 go down, there's more free available miR222 to have those beneficial effects, and there's also a decrease in DCHS2 transcription, so more of the YAP is retained in the nucleus, and it has potentially those beneficial effects, as we showed. So just to kind of encapsulate some of those points, I think the main take-home messages are that the heart obviously grows in response to both exercise and pathologic stimuli, but the outcomes are very different. So we've been interested, as others have been, in understanding what the basis of that differences is. And at least in our hands, when we look at these transcriptional profiles or proteomic profiles, they're really very different at an early stage when the hearts really look superficially very similar, suggesting to me anyway that there's a fundamental qualitative difference that occurs early, setting these on different paths. And we would argue that exercise provides a platform for trying to figure out what these beneficial pathways are that is complementary to the study of disease, because there are distinct pathways. And in particular, we've talked about link-exact1 as a potentially tractable target, since there are FDA-approved drugs that are essentially antisense reagents, and we would argue this might be something that could be mimicked to try to inhibit link-exact1 and recapitulate some of the benefits of exercise. And then in the last couple of minutes, I guess I would just focus a little bit more philosophically on the study of exercise. And I would say the most common question I get asked when I talk about exercise is so with all this hard work, when can you tell me that I can take a pill that will make it so I don't have to exercise? Obviously, that is not our goal. Somebody in the lab did find this on the internet. They're selling exercise in a pill. This is not an off-label recommendation. I can't imagine this actually does anything. It's calcium pyruvate. And so for those of us who can exercise, we should. And so obviously better to take the stairs than the escalator to the fitness club. And as a clinician, my philosophy is kind of summarized here. Everyone's too busy to exercise, but would you rather exercise an hour a day or be dead 24 hours a day? As commented here, our society has obviously become progressively more inactive in many ways, and this coincides with the metabolic stresses that have come in recent years as well, which is probably a double whammy. And so I do think obviously moving is beneficial. I do have a walking treadmill desk, which I don't know if it's doing much good, but certainly makes me much happier. I have not, however, resorted to more ambitious ways to be less sedentary. I don't think I'm capable of doing that. And some of these recommendations, I think, are probably misguided. This one seems a little bit extreme, especially when you think about the whole, water and electricity thing going on. The other question I frequently get asked is, how much exercise do you need to do? And obviously, it depends what your goals are. If you want to be an elite athlete, a lot is the answer. But if your goal is to promote cardiovascular health, one of the encouraging things is it takes relatively modest amounts of exercise to have an impact. And so as many of you know, at least based on the observational data, going from sedentary to really minutes a day of exercise have a significant impact on what are termed preventable deaths per year. And this was picked up by the New York Times recently, as just saying 10 minutes a day may lead to a longer life, although obviously you can see you're still on the ascending limb there. And so there's still more benefit to be had. And then, the other question is why is exercise different? Cardiologists frequently say to me, if I have a heart attack, isn't the rest of my heart just exercising more? And so why is it different? Of course, it's always hard to answer the why questions. But I would point out that there's probably been a strong evolutionary pressure to have a a beneficial response to exercise. It's how our ancestors survived, and really no selective pressures on things like heart failure or myocardial infarction that generally don't affect reproductive success. And my friends who are true physiologists point out that, the mechanics and the physiology are not exactly comparable depending on what kind of exercise you're talking about. It's not really the same. But my bet is more that it's not all hemodynamics, that circulating factors and some of the non-coding RNAs we talked about today do circulate. I think we don't have any direct evidence that's the circulating portion that's important here, but we've also been interested in proteins that circulate and small molecules that may also have an important effect there. And in that context, I do think it's fascinating that Rusty Gage and others for years had a prior literature that exercise is one of the stimuli that can induce neurogenesis in the adult brain. And now we, I think, demonstrated that it's, so far the only one that we've identified that induces cardiomyogenesis. Are these discrete, distinct responses, or is there a common signaling pathway that does this, or even potentially circulating factors that are playing into this? And so we're quite interested in identifying those common biological pathways that may be a clue to how to have the beneficial effects of exercise on a broader scale. So most importantly, let me just emphasize again the people who really did all this work. Hao-Bo Lee is in the center there, again, remarkably talented instructor who really championed all the LinkExact-1 work. Xiaojun Liu had done the MIR222 work and is now a scientist in industry. And Carolyn Lerkenmuller did and continues to work on some of the cardiomyocyte proliferation study with Rich Lee. She's now an independent investigator back in Germany. So again, thanks so much for the invitation. I would hasten to point out we benefited from lots of wonderful collaborations. Juan Ma Gonzalez Rosa, I pointed out Mike Bristow, John and Cricket Seidman, who have been long-term mentors and collaborators and shared the MHC mutant mice, and then Rich Lee's lab, as I mentioned. So thanks so much. I'm delighted to answer any questions. Maybe I'll just stop sharing the screen so we can see each other. And
thank you so much, Dr. Rosenzweig. So I was just about to cancel my Peloton membership and inject myself with some LinkExact-1 inhibitor, but thank you for that perspective at the end. Not an exercise pill yet, but we have several questions in the chat, and I want to invite the rest of our audience members to continue to type your questions in the chat, and we'll try to get to as many of them as possible. And the first question I'd like to ask is from Dr. Dale Abel, our new chair of the Department of Medicine. And he says, welcome to UCLA, Tony. Do you have any insight into the regulators of the link RNAs that are induced by pathological or physiological hypertrophy?
Yeah, so thanks so much. First of all, Dale, wonderful to at least see your name in the chat, and hope we'll connect soon. Congratulations on your new role. I know in my conversations today, so many people are so excited about you being at UCLA. So that's wonderful. I'm sure you'll do great things. We have done a little bit on the upstream regulators, particularly the physiologic. And so, as you will not be surprised to learn, LinkExact-1 appears to be regulated by PI3 kinase and AKT signaling, two of your long-term favorites, downstream of insulin and IGF-1 signaling, and so they will inhibit LinkExact-1 expression, and I guess that is most of what we've gotten, and it does look like that will happen at least in vitro models in response to the growth factors that we know increase in exercise, like IGF-1 or neuregulin, things like that. So that's kind of as far as we've gotten in that state. It is interesting, as you know, but not everybody may know, AKT is activated both in physiologic growth, where it's necessary for physiologic growth, but is also activated in pathologic growth. And so interestingly, though we don't, there's another layer of specificity there that we don't explain, and that this relates to the work that Tony Muslin published years ago looking at isoforms of AKT. So pharmacologically and with vectors, AKT and PI3 kinase activation inhibit LinkExact-1, but that doesn't fully explain the difference, as you'll appreciate, given that we know it also is activated but has very different effects in pathologic hypertrophy.
Our next question is from Dr. Marlene Tuma, and she asks, did you evaluate link exact 1 expression in the plasma of pregnant women with physiological hypertrophy or the athlete's heart?
We haven't really done much along these lines with pregnant women. We have separate projects where we've been quite interested in pregnancy, and we have looked at some models of pregnancy in mice. And, you know, as on that Joe Hill, Eric Olson schematic, pregnancy and exercise tend to be lumped together. Actually, what we see, which I think resonates with work that Leslie Leinwand had published years ago, pregnancy is pretty different from exercise. And so we're not, I think that's a different box. So at least based on, this is mostly based on cardiac profiling. And again, Leslie Leinwand had published things along these lines a while back. So I think there's more to the pregnancy story, and it's a little confusing. And from our perspective, for example, some of the things that we see as physiologic markers like CBP beta going down in exercise heart actually goes up in pregnant hearts. And so I think there's more of a mixed bag in pregnancy, but we haven't had the opportunity to look in plasma of pregnant women. And then in athletes or athletes' hearts, you know, I guess the good news, bad news is, you know, thankfully we don't get a lot of those. So we haven't really had the opportunity to look. I guess the prediction would be that it would go down, you know, and so we haven't had hearts from athletes where we should be able to measure it. Plasma is tough because the levels at baseline are pretty low, and so we use digital droplet PCR to measure, and we certainly see an increase in heart failure patients. I don't think we were able to detect a decrease from the low basal level in the plasma of athletes, but all those things would certainly be interesting, but not so much opportunity so far. But thanks so much for the question.
And next we have a question from Dr. Tamer Salam, and he says, great talk as always. First, 70% sequence similarity is really strong conservation for a link RNA, which is exciting. Do you think some of the non-coding transcripts may encode a micropeptide?
Yeah, so it's a great question. And I've got to say, we've worked pretty hard to try to see in a couple of different ways, you know, first, are there any predicted, you know, reading frames? And then, you know, can we see whether there's anything being transcribed, you know, by taking those and seeing if we can detect protein expression, or, you know, taking upstream sequence and hooking it onto something else, you know, GFP, and seeing. And so far, we don't have any evidence that there's a micropeptide that's encoded in link exact 1. So we haven't, but we were curious about that as well, given, you know, a number of really elegant studies by Eric Olson and other people showing that some of those micropeptides have really profound effects, and that would have been a cool result. But we've been unable to demonstrate that, at least of the open reading frames that we've seen, that any of them are actually, you know, translated into protein.
And Tamer has another question. It's wondering if you can comment on the stoichiometry of how the link RNA functions, and is there enough link RNA link exact 1 to buffer miR-222?
Yeah, it's a great question, and you're much more of an expert than I am. We have tried a little bit to do that with trying to, you know, have standard curves and quantify, and it's hard to know. I guess I would say in our hands a little bit, but I think, you know, depending on the cardiomyocyte and again, mostly looking in the mouse models, you know, we do think we're kind of in the, you know, from a low of sort of dozens of copies to a high of hundreds of copies, and you know, back of the envelope calculations comparing to what other people have done, we think that might be in a range where you could flip this switch back and forth, but I've got to say we struggle with that. It's also split between the nucleus and the cytoplasm, and so we don't have precise numbers on which compartment it has, and so whether that would be, you know, sufficient to buffer miR-222, I guess, is a little bit hard for us to say, depending on the induction of miR-222. So yeah, I think it's a really interesting question that we don't have we've struggled a little bit, but don't have a precise answer for, but we think we're kind of in a ballpark where it could have effects. Of course, as illustrated, we don't think miR-222 is the most interesting part of the story, but we do think it could be part of the story just because it does seem to bind it. And based on the in vitro data that I didn't show you, it does look like miR-222. If we inhibit miR-222 downstream of the knockdown, we kind of reverse some of those effects. So it looks like in cardiomyocytes, it does have some, but those are all, as you would point out, kind of artificial overexpression and knockdown experiments, not the physiologic state.
I think we have time for a few more questions. One is from Dr. Katya Bravo, one of our adult congenital cardiology fellows. And she says, thank you for this great presentation. Has LINK-EXACT-1 been found to mediate disease processes in other conditions where LV hypertrophy predominates, such as coarctation of the aorta or hypertrophic cardiomyopathy?
Yeah, great question. We, honestly, we haven't had a chance to look. If you have samples, we could send you sequence, or we could do the PCR. We'd love to know. Yeah, I think that would be fascinating to know. I guess based on the animal models, I would predict that something like coarctation, just like constriction of the aorta in our animals, would induce it. That would, but I don't know the answer. And then, I think hypertrophic cardiomyopathy is really interesting, right? Because presumably that works through different mechanisms, and so I think that would be a fascinating thing to look at if there were samples available. And obviously the Seidmans have generated HCM mouse models, which to some extent recapitulate the biology, and we haven't looked at those yet, but it's a great question.
And I think the last question we have time for is from Feng Guo. And the question is, how much is known regarding how exercise increases miR-222 and how pathological conditions increase LINK-EXACT-1 and DCHS2?
Yeah, so I kind of mentioned only a little bit, is known about those things. So and the place we looked first, because it's something we, like Dr. Abel, have been interested in a long time and is so integrally wrapped up with the exercise response, is PI3 kinase AKT signaling. And so for sure, that's part of the story. And so it does look like those things induce that some of these changes are downstream of those. And certainly, so when we inhibit PI3 kinase, for example, PI3 kinase 110 alpha, we see an increase in LINK-EXACT-1, which may be part of it in vitro. But beyond that, and how things like mechanical forces and stretch and heart failure and what the mediators are all really interesting questions that we don't know the answer to. I guess at this point, I would say, and when we overexpress or activate PI3 kinase AKT, we inhibit them and recapitulate the kind of expression that we see in the mouse models of exercise. So we think that's part of the story, but I'm sure there's other elements that we haven't identified yet.
Well, thank you so much, Dr. Rosenzweig. That was a terrific presentation, and this is really exciting data. And I think this is opening up so many different exciting opportunities for investigation. I really love the study of exercise and the beneficial effects of exercise on cardiac development and cardiac remodeling. So this is really terrific. Thank you for visiting virtually UCLA, and we look forward to having you physically on our campus one day, and hopefully not the too distant future. So
no, thanks so much. Lots of fun. And really thanks for all the great questions and for the opportunity to present. I would say we don't know about the combination of LINK-EXACT inhibition and Peloton. So, you might try both. It could be synergistic.
Sounds good.
Okay.
All right. Thank you, Tony.
Thank you so much, Jeff.